So, all the enzymes that you've used these couple of weeks had to be discovered at some point in time, right? And so, the way I think about enzyme discovery and workflow discovery is that really nature has discovered every enzyme out there to do every transformation of DNA or RNA or protein. Really has discovered every workflow. So if you can go into nature and understand how nature copies its DNA from one end to the other, or modifies its RNA, or degrades protein, how does nature do that? If you can really understand how nature does it, then you have a shot at really discovering enzymes from nature by asking these fundamental questions. And then once you understand how nature does it, then we can do it even better and improve those enzymes. Okay. So there are a couple different platforms that we use to discover enzymes. One is called functional genomics. And really this is the idea that in every scoop of dirt, in every drop of water, there are microbes and there are organisms that are living in that specialized environment. And so if you scoop up a teaspoonful of dirt, there's millions of microbes of fungi in that sample of dirt. And in each one of those organisms, in their genomes, there's a huge catalog of enzymes, genes, and enzymatic activities. So by scooping up dirt in different environments, you can extract the DNA. And by extracting the DNA, now you have access to all the different genes and all the different enzymes that are in there. So how do we access those genes and enzymes from these environments? One of the ways that we do that is to take the environmental DNA that was scooped up and purified, and then we've flown it into what's called a phosphate vector. So each one of these fragments here is about 40,000 nucleotides and contain maybe 40 different genes. So then we can take this environmental DNA and then put it in our favorite E. coli, and the E. coli will express the genes from the environment. So now you have an E. coli with an insert that's expressing all the genes that were in this environment. And then the fun part comes in, is trying to figure out which of these E. coli are actually making the enzyme that you want. And the way we do this is that then to sometimes put each one of these E. coli into a different well or plate, and then design an enzyme assay or a colorimetric assay to search for the enzyme enzyme function that we want. So this is a way of searching for enzymes by looking for the actual function via an assay. So maybe it's an enzyme that cuts a particular substrate and then turns that substrate from white to blue, for instance, or it's an enzyme that will cut DNA in a specific way. So if you have a good experiment to actually test it, you can go out and find new enzyme activity. So the other major way that people use to find new enzymes is bioinformatics. So in databases these days, people are sequencing tons and tons of organisms, tons and tons of environments, and really getting a huge amount of DNA sequence in databases. And really the trick is to figure out which ones of those, to try to predict the function of these DNA sequences just from the DNA sequence. There's a huge challenge. Do we have any real bioinformaticians in here? Again, the idea here is that you have a DNA sequence here, and you can say, okay, we see 4 genes here, but how do we actually use bioinformatics and computational methods to actually predict the function just based on the sequence? So that's a real challenge. So this is sometimes useful. You can say, okay, this one, the sequence looks like a DNA polymerase, for instance. So you can say, okay, we found 10,000 DNA polymerases, but we don't really understand the differences between those polymerases just on the sequence itself. But this is a really ripe area for discovering new enzymes and new related enzymes just from the DNA sequence. And a good example here is really trying to look into those databases and create tools to try to cluster together sequences of DNA that look similar and different. So this is an example of this enzyme called TET, which is an epigenetics enzyme. And each one of these dots is a different protein sequence here. And you can see here that this TET is one of the ones we know about, so we know its biochemical activity. And in this circle here, these are all the ones that the amino acid sequence, they look pretty similar. But what you also see that there are other clusters of similar sorts of proteins that might have the same activity, but they might not, right? So you have all these other sequences that look kind of the same, but we don't know anything about it. So right now, this is a major challenge in the field, is that you have all this information about all the different sequences, but you don't really know the underlying activities that much at all. So there aren't very good methods to test the diversity of the sequence here. That's one of the really big challenges. Okay, so one of the interesting things in biology too is that enzymes in nature don't really work by themselves, right? So in many natural pathways, enzymes are handing off a substrate from one enzyme to the other, to the other, to the other. And so we don't really understand which enzymes are included in this pathway and how the directions of the genes might affect things. But there are ways of assembling these, and we'll talk about this using robotics. And more and more people are trying to use robotics to take the place of just putting things in tubes. Okay. The other discovery platform is really high throughput protein engineering, and so this is where if you have an enzyme that you're interested in, but it doesn't quite have the activity that you want. So maybe it's an enzyme that works at 37 degrees, but you actually need it to work at 4 degrees. And so there's a lot of effort going into how do you actually engineer or change the protein to have the characteristics that you want. And so AI is really dominating the conversation here, and there's a lot of companies that have started up that are using AI to go into the databases and try to predict how to mutate the protein to actually change its function. So you might want a protein that has higher activity or higher or lower temperature. It might not be compatible the buffer that you actually want to use it for, or it might be insoluble. You might need to make it more soluble. So really trying to use AI to solve some of those problems. So again, in the state of the art field right now is that the AI is way ahead of actual testing the enzyme. So that the computer people can really predict what they think is going to be important, but the slow step is really making those proteins and testing. So, some of the places where we sample, every time we would go travel, we'd scoop some dirt, and we'd go in the fields, go hiking, and scoop some dirt there. So every teaspoon of dirt has millions and millions of microbes. And to me, I look at those millions and millions of microbes and say, okay, that's millions and millions of potential genes and interesting enzymes. So we're really scooping up some dirt from the mountains. Biology is near the ocean, and so we do some work in salt marshes, and the oceans are a really vast and interesting area of enzyme, microbial and also enzyme diversity that's really untapped. Okay, so, yeah. Okay. Yes, okay. So my research was on hot springs microbiology and hot springs DNA replication and repair. So this is a great map of all the hot springs areas around the world, down South America, Asia, up in Russia, in the Western U.S. So why do we want to look at hot springs microbiology, hot springs enzymes? Stream of files, yes, exactly. So PCR, right? So just to remind everybody, that very first step of PCR is to separate the two strands of DNA, so then you can anneal those primers. So it was realized early on that, okay, this is a big problem, and that the enzymes that are in the PCR have to withstand those really high temperatures. The first kind of methods of PCR was actually using plenal from E. coli and doing the same cycling, but they'd have to replace E. coli enzyme every single cycle. So someone realized that, oh, there's actually a enzyme, Taq polymerase, from a hot springs at Yellowstone that had been discovered in the 70s that actually withstand those temperatures. So that was a huge leap forward. But again, thinking about you have a molecular problem, you have a technology problem, you need enzymes that work at high temperature, and then turning to nature to figure out how nature does it. So again, these are all the thermal sites around the world. And this is a video taken by the Alvin in Woods Hole. Has anybody ever been over to taking the ferry to Martha's Vineyard? Yeah. So it's so as you take the ferry from Martha's Vineyard and leaving Woods Hole off to the right, sometimes the research vessel is in port, and they have a deep sea submarine right on deck. So it goes out of Woods Hole, goes around the world. This was taken in the Gulf of Mexico at a hot spring hydrothermal event by the Alvin. And so this is an amazing feature here. So right in the middle here, there's superheated water that's percolating up through the Earth's crust. And so right in the middle here is about 300 degrees centigrade. It's super, super hot. And the water is around 4 degrees, super, super cold. And so when the superheated water percolates up, all the minerals precipitate out and form these beautiful cones at the bottom of the ocean. So what's really interesting is that right in the middle here is 300 degrees, but then it's cold. So there are these tremendous temperature gradients from 300 down to cold. So Woods Hole broke off a little piece of this smoker and brought it back, and we were able to isolate organisms that live at about 100 degrees in this cone. So it's amazing that organisms can live in boiling temperatures, and not only can survive these temperatures, but actually require these high temperatures to actually live and grow. So kind of an amazing spot. So again, this was a really fun part of the job, is to say, okay, let's figure out how nature does this, and let's go sample in a lot of these hot springs from around the world. So has anybody been to Yellowstone National Park? Yeah. If you haven't, it's an amazing spot, and of course a hot spot of geothermal activity there. So this is Octopus Spring, and this is where Taq DNA polymerase comes from. And you can see here, just by the colors, that right in the middle here is about 85 degrees, 85, 90 degrees, and there's some light here, but then as the water flows out, it cools down, and then the colors change here. So Thermus aquaticus is about from right here at about 75 degrees. And so this is the Octopus Spring where Thermus aquaticus was originally isolated. So if you go on a tour of the hot springs with a park ranger, they'll always make the point that yes, this is where Thermus aquaticus was first isolated. This was the origin of the Taq DNA polymerase. Taq DNA polymerase sells for probably $1 billion a year around the world, and Yellowstone got screwed. So now when we go to Yellowstone to collect samples, we first sign an agreement that says, okay, if we discover anything from this really unique natural environment, then we'll give a royalty back to Yellowstone National Park. So I think that's yep.
Is Thermus aquaticus bacteria, or what is it exactly?
It's a bacteria, yes. So that's one of the other interesting mysteries here. So it's a bacteria, and if you look at the proteins look are those proteins? Why are they active at high temperature? And so there's been a lot of active research about that. The bottom line is that there's no easy answers. Some people thought, well, maybe they're more disulfide bonds or something, but it seems to be these high temperature proteins are thermostable because of lots and lots of small little interactions that have evolved over millions, billions of years. So again, in these hot springs, what's amazing is that they might seem like a very sterile and extreme environment, but in every teaspoon of water, there's about 1 million microbes that are living there. So, an incredible source of enzymes. The other thing about octopus spring is, well, why they focus on the spring? And what you don't see in this picture is that the road is right next to it. So, it's an easy to study subject. So that's why I did it back in the 70s. This is another one that's an amazing mud pot. This is a mud pot with these bubbling like hydrogen sulfide coming out. This is pH 1, believe it or not, and it's about 80 degrees. So that's an amazing extreme, right? So, but in every scoop of mud here, it's about 1 million cells. So again, these organisms have evolved to this super acidic environment, and again, not only they're surviving in this environment, but they've actually evolved that they use extreme conditions to actually live. So this is kind of another acidic mud pot. You can see dry sulfur up here, and it's just like boiling cauldron of craziness. So it's amazing. Again, more mud pots. This is a 73-degree sample, and you can see that there's this thick mat of light. So again, just incredible. This is almost 60 degrees, and it's you kind of see from the pH paper, but it's an incredibly acidic stream. So you have really nothing living on the edge here, but then you have these mats of bacteria just growing and thriving. Yep.
The question, when you
Yeah.
collect from the bottom of the sea,
Yes.
do you take it to the source? How do you keep it? In the boiling water in order to maintain pressure or?
Yeah, so we don't really worry about the pressure, and if we're going to grow the organisms, then a lot of those conditions are anaerobic, so we put them in an anaerobic vessel for fermentation. But also we figure that going from those high temperatures, even to room temperature, that drop of 60 degrees is like putting the organisms in the refrigerator. So we found that they're fairly stable, just because we're lowering the temperature and they're slowing down the metabolism.
In the case of the one species or one,
Yeah.
you have it in the acidic environment, do you collect it for human growth?
So the media that we grow is acidic. Yeah, so we can alter the pH to grow in those acidic conditions.
But all the organisms that you collect are cultured.
No. So it's estimated that 99% are unculturable. So we do a mix. We try to culture them, and what we can culture, we culture. But then the other 99% we just extract all the DNA, and then have access to the actual genes and everything. Yeah. So I think, the things that you can culture is really the minority of what's out there. So it's a real challenge to invest the time to try to make these things grow. It's so for this one, we don't know what other cofactors it needs to grow. So here we just scoop it up and extract the DNA. So there are hot springs in Europe too. This is one in the Azores, which is some islands in the middle of the Atlantic. These are much more civilized hot springs where they build lots of baths based on the hot water runoff. But we would sample along the way there. That was a beautiful spot. This is what's called hydrogenobacter, and this is hydrogen bubbling up, and you can see this white mat here is really feeding on the hydrogen as an energy source. Yeah. And that's a blob of microbial life. And then over in Russia and Asia and Taiwan, there's some hot springs right outside of Taipei, and that have these are acidic boiling hot springs here. And I think the sign said don't go over there and sample, but we did. This is an example of an organism called Sophilolus sophocaucus that lives at pH 2 at about 80 degrees. Yep. So we had an amazing trip to the Kamchatka Peninsula on the far east side of Russia, and so it's this peninsula that's north of Japan, where they just opened up to it was close to the world because it was such an important naval port in the late 90s, and so this is an amazing volcanic area, and there aren't really many roads, and so they would take us around in helicopters, and the whole peninsula is dotted with these volcanoes and hot springs there. So here we are getting off this helicopter in this beautiful valley with the hot springs, and then they fly away and hopefully we came back. But this is like one of the calderas, they call it, all with hot springs, and all around the caldera were blueberry bushes, really kind of wild place. And here we are sampling some bubbling hot springs there. And again, I'm just taking a little bit, putting it in the test tube, and again, just a couple mils will provide millions and millions of cells. So I said there are blueberry bushes around hot springs, and so there are also these huge chocolate bears around. So you kind of see my foot, but the prints were huge. One of my other favorite examples that from hot springs was this Italian island called Vulcano, and this was before my time, unfortunately. But this is an organism that was isolated from this beach here. So low tide, the tide was out, and it reveals all these little boiling hot springs right at the beach. And so they scooped up some mud here, brought it back to the lab, and cultured this organism called Thermococcus liberalis. And from this organism, they cloned the DNA polymerase called bent DNA polymerase, and this was really the first proofreading PCR enzyme that was available. So it's really kind of from this beach, from that organism, was really the birth of proofreading polymerases for PCR. Okay. So in traditional gene cloning, if you're interested in a particular gene, what you have to do is get the organism or the sample. You'd have to extract its genomic DNA, and then you'd have to design PCR primers to amplify that gene specifically. Right? So you'd amplify that gene specifically with PCR, and then you would clone that gene into a vector and express it. Okay? So if I wanted to, say, clone a low temperature DNA polymerase from a king crab, I'd have to go get that king crab, and then I have to grind up, and for eukaryotes, you grind up the RNA, convert it to cDNA, and then clone its gene, and clone it into an expression vector. So the basis of all this is you have to have the crab to start with to find the crab genes. So what's completely changed is the concept of gene synthesis. So now, instead of getting that king crab to clone its DNA polymerase, you would go to the king crab genome on the government websites, find the protein that you're interested in, and then you convert that protein into a gene, and then you type out that gene on your computer, send it away, and a few days later that gene would arrive. So this concept of gene synthesis really cut into my travel time for going to hot springs. So now instead of going to a hot spring in Yellowstone or Kamchatka or Italy or Taiwan, wherever, you go to the hot springs database of sequences and find all the different DNA polymerases or enzymes that you want from that actual database. So it's much less exciting than the travel we did, but it's, you know, you have access to thousands and thousands of high temperature genes. So really the idea there again is gene synthesis, and this has really changed the depth and the breadth of how people can actually clone and really understand genes. So the idea here is that you can go and find a protein that you're interested in, and usually you start with the protein sequence, and then you can use just simple web tools that convert that protein sequence into a gene sequence. And then the way the gene synthesis works is that the companies actually break this long DNA sequence into short oligonucleotides that overlap here, and from the gene synthesis, you actually get a piece of DNA that corresponds to the gene that you can then make protein. Okay? And this is super cheap now. It's about, you know, just a few cents per base. So this has really changed how people actually clone genes and discover genes at all levels. And one of the ways that they make oligonucleotides for gene synthesis is by chemical coupling. So this technology hasn't really changed in many years. And so it's a chemical process where they build the oligonucleotides block by block on a solid support by adding what are called phosphoamides and adding A, and then, you know, that are blocked, and then adding the next nucleotide over and over and over again. So now you can build up these oligonucleotides to about 60 nucleotides without a problem. They can go longer than that, but then the error rate becomes a problem. So again, like chemical synthesis, you can synthesize the oligonucleotides, let them overlap, and then that's your gene. So this is a really chemical heavy process that creates a ton of waste, and you have to have really specialized facilities to do it. What people are also really interested in is using enzymes to actually make oligonucleotides in the same sort of way. So here, instead of using chemically modified nucleotides, you can use special nucleotides in a DNA polymerase to actually build up a single-stranded piece of DNA just by adding nucleotides to the end. So there are companies that are trying to do this, and they're arguing that they can actually make longer pieces of DNA. They can make pieces of DNA that are more accurate and faster and in a more clean way. So again, enzymes are a biological process, and really there's not a whole lot of chemical waste here. Okay. So, any questions about how we discover enzymes? There are so many enzyme activities out there that I think every one of us could, you know, spend a career looking for different enzymes and still not go through them. So it's a really fun thing to do. So once you've discovered an enzyme, really the next step is how do we actually go about and make enzymes at scale, the cell. And because of this course, you have a good foundation about some of the basics of cloning and purification, so we'll go over this a little bit. But really the process is like a lot of what you've learned. So really the complete picture is first you clone that gene of interest into a plasmid using gene assembly or restriction enzymes, and then you transform E. coli or yeast or another organism with that plasmid, grow them on a plate, and once you confirm that, then you take a single colony and grow them in liquid media, and in that liquid media, those bacteria are going to be making the proteins that you want, ideally. And And then after you blow them up, you lyse them open, and in those cells, the next trick is really to separate the protein of interest from all the other thousands and thousands of E. coli proteins that are come from the cell. And after purification, you're just left with the protein of interest. So at biolabs and other companies, there are teams of people who are really good at cloning, really good at protein expression, really good at purification, and then spend their careers getting super good at each of these steps. So together, there's a really high efficiency, high success rate. Okay, so this should be familiar to you, right? Gene cloning, really the basics is having your insert, putting it in a vector to create a recombinant molecule that will produce the protein that you're interested in. At biolabs, we would almost all the enzymes that we make were expressed in E. coli, and E. coli is a really great model organism to actually make proteins. And why E. coli? And E. coli has been a real model organism in the lab for many, many years, and the genetics are really well understood. So if you need to change the underlying strain of E. coli, that's easy to do. The other great thing about E. coli is it grows really rapidly. So it doubles every 20 minutes. So if you're growing up E. coli, you don't have to wait a long time. E. coli is efficient, it's cheap, but the drawback of a bacterial system is that if you're trying to express a eukaryotic protein, then it can be a little bit more problematic because it doesn't have all the machinery that eukaryotes necessarily do. Okay. So I just wanted to mention also just patents in all of this as well. So patents are certainly an important part of biotech, and patents in its most fundamental form protect an invention, and by getting a patent that encourages people to invest in development of new technologies if they're able to protect and patent them. So there are really two approaches for patent protection. Getting a patent will exclude others from using your technology, and you can develop it yourself, so you have a monopoly on your technology for about 17 years. Or you can get a patent and then license that technology to other people who can then use it and then give you a fee for that technology. So back in the 80s, people really didn't care about patents, and some people still don't really care about patents. But in 1980, really no one patented living things until Stan Cohen and Herb Boyer at the University of California patented the method of making a recombinant protein by putting one piece of DNA into another piece of DNA and expressing it in a third organism. So they had this patent in 1980, and the Stanford and UC system licensed that out to any company that was making any protein that they would sell. And so this patent generated about $300 million in licensing fees to Stanford and UC. So patents are a big part of biotech, but nowadays universities are more and more interested in getting patents and licensing the patents to generate revenue. And who knows what's going to happen in the funding situation now, but certainly universities are going to be trying to squeeze as much out of patents as they can. Yes. But there's some also interesting things about patents. You used to be able to patent a naturally occurring protein. So back in the 80s and 90s, biolabs would actually patent restriction enzymes coming from different organisms. And then the Supreme Court said you actually cannot patent a naturally occurring thing, which kind of makes sense. So all those patents were invalid. So no one can patent an actually naturally occurring thing. So if you had a really interesting enzyme that you discovered from a hot springs microbe and you wanted to patent it, how would you approach that? How would you try to get a patent? Process. Yep, patent the process for sure.
Or just trade secret.
Or this trade secret, right? So some people say, okay, in this tube, we're not gonna tell people what it is, but we're not gonna patent it either. So there's risk there in the trade secret that someone will reverse engineer it, figure it out, and maybe patent that, and then exclude you from it. Any other ideas of how to, yep?
Could you patent effectively use the problem?
Yep. So that's not naturally occurring, right? So you can use the vector that you use. Any other ideas of how you might patent it?
You could add a synthetic element and patent that.
Exactly, right, so you can take that naturally occurring, say, polymerase and introduce a few mutations, and now it's not naturally occurring engineered, right? So you might want to for that. So that's typically how we approach it. We might find something that's naturally occurring and have one scaffold, and since we can't patent that, then we do a little engineering to improve it, but also to be able to patent that actual sequence. Yep. Okay. I think I'm gonna stop there. You can do that, you can do